Hi everyone, and thank you for joining today's webinar. My name is Fanny Sandberg, and I'm a Management Associate at the Swedish American Chamber of Commerce in San Diego. And for those of you that aren't familiar with our organization, we promote trade and facilitate business opportunities between Sweden and the US. I also want to add that we are a part of the Life Science Bridge together with chambers in San Francisco and New England. Through Life Science Bridge, corporate members such as Getinge can get help from an organization with extensive network and a local presence in order to expand business opportunities further. If you want to know more about us, I have sent you all the information in the chat, where to find us and where to follow us. Before we start, if you have any questions, please write them in the chat function. You should see it at the bottom of your Zoom window. Then we will answer them during the presentation. In today's webinar, our speaker, Anne D'Ambroso, Manager of Product Applications at Getinge, will talk about how Getinge brings passion for life with its bioprocessing product line. Welcome, Anne, the floor is yours.
Thank you so much for having me, Fanny, and for that introduction. Today, I will talk to you about how Getinge brings passion for life with its bioprocessing line. Oh, I have too many things open with the chat. There we go. So Getinge was founded in 1904 in the small town of Getinge in Sweden, and I apologize for my lack of Swedish pronunciation. But we have sort of Americanized the name here. Nonetheless, this company has more than a century of experience, and it's rather large. In 2021, net sales were over 30 billion SEK, or approximately 2.8 billion in US dollars. 90% of our sales go to hospitals, and the remaining 10% is in life science institutions, such as pharmaceutical and biotechnology companies. We have approximately 25,000 customers in more than 125 countries worldwide. The headquarters is located in Gothenburg in Sweden, but there are operations in more than 35 countries. And more than 10,800 employees are located all around the world, working with everything from R&D and production to sales and service of the products and solutions that Getinge provides. It is our firm belief that every person and community should have access to the best possible care, and we empower our customers with knowledge, technology, and resources needed to set up the most efficient structure to save lives. Through advanced life-saving technology, infrastructure design, and workflow expertise, our goal is always to improve the personal experiences of patients and professionals within hospitals and different life science institutions. We are working together with our customers with 5 different main categories for improving clinical and economic results. You can see those here. We have intensive care, cardiovascular procedure, operating rooms, and sterile reprocessing, and life science, which is where my role lies. So today I'm going to focus on the life science part of Getinge's technologies. And in fact, my specific background is that I came to Getinge almost 3 years ago with the acquisition of Applikon Biotechnology. Applikon has been making bioprocessing equipment for almost 40 years, making us a very strong partner for Getinge to grow in the life science market. And aside from the growth of sales, Getinge is intent on growing awareness about the life science business. And with that intent came a campaign centered on the idea of bringing science to life. So with a focus on contamination prevention and upstream bioprocessing, we provide tailored solutions to scientists and engineers who are dedicated to preventing, mitigating, and curing diseases to save lives, and also working in different applications that support innovation of other technologies. And together, all of these areas are supported by the strength of Getinge as a company, with our solutions and worldwide presence, our expertise, and our service team. And the variety of ways that we work and support our customers is really one of the reasons that I find this job to be so fulfilling. And I'm very excited to share information about our work with you today, and hopefully you'll find it as exciting as I do. So the infographic that I'm showing here is one way of looking at a path for research and development that occurs in creating a clinical therapy such as a biopharmaceutical. There are 3 life science product lines supporting this: cleaning and sterilization, bioreactor systems, and aseptic transfer. And I really am focusing on the bioreactors today. That's my area of expertise. And you can see that the 3 core parts of this workflow, the research labs into clinical trials and production, are going to rely on bioreactor systems for some of their most critical work. So I hope I can help you get excited about how modern science and how bioreactors are helping biopharmaceutical development in related fields. But I recognize not everybody really knows about bioreactors, so we'll start with the first question: what is a bioreactor? A bioreactor is a chamber for growing organisms, could be bacteria, yeast, or mammalian cell cultures, and the goal is to have them under controlled conditions. So you can already get some clues using the pictures here about the key features. First, the vessel is meant to cultivate a single type of cell. So if a researcher is conducting an experiment, they don't want other types of cells to contaminate that experiment. And the particular cell line they want to grow usually has an optimum for pH, temperature, and oxygen level. So the fundamental purpose of the bioreactor is to measure and control those key physical parameters in the chamber so that that bioreactor has homogeneous conditions throughout, and it is maintaining an optimal set of conditions. So yeast, for example, grows fastest at 30 degrees Celsius and a pH of 5, whereas human stem cells require a temperature of 37 degrees Celsius and a pH more like 7.2. The maybe more interesting question is, what can be achieved by growing cells in a bioreactor? Well, biotechnology has enabled production of many interesting products: biopharmaceuticals, antibodies, vaccines, also for bioconversion of organic waste. But right off the bat, some of the images on the bottom left of my slide might catch your eye as something that's familiar. In fact, combining grains or fruits together with yeast creates fermentation, and containing this in a vessel is one of the oldest forms of a bioreactor. It produces some of our favorite things: bread, beer, wine. In the 16th century, Germans created a purity law for beer and defined it to only include water, hops, and barley malt. But that law was updated in 1906 to acknowledge that yeast cells are actually a key ingredient. A modern clinical example of the progression of science is with regards to diabetes. Type 1 diabetes was fatal until it was discovered that insulin extracted from a cow or a pig pancreas could control blood sugar levels. In the late 80s, scientists genetically engineered a strain of bacteria which could be cultured in the bioreactor, and it produced an exact copy of the human insulin molecule. And this human insulin molecule had a higher purity, so it avoided allergic reactions, which were still quite dangerous to some patients in the clinical setting. So today, there's already an even further improvement on that, which is the goal of culturing stem cells in bioreactors, which might be injected directly to a diabetic patient so they could grow their own new pancreatic cells to make insulin again. That's just one example, and you may be asking yourself, how is this possible? So I'll take one more step into some of the basic building blocks of biology as an explanation of how this works. So hopefully you're a little familiar with DNA and its double helix with A, T, C, and G codes. And after COVID-19, I'm sure you've also heard about mRNA, or messenger RNA, which is a translation of the DNA codons. RNA codons together are encoding for amino acids, and amino acids together combine to make proteins. Proteins function in a sequence to carry out metabolic reactions or biochemical conversions of different molecules, and that series of reactions is contained within each cell. If you put cells together, you get tissues. So having this fundamental understanding of the building blocks has really enabled some fascinating science to progress, and scientists are using these different pieces to either modify or create new versions of proteins, of mRNAs, molecules that can give improved functionality for new applications. Some more examples, again, DNA can be packaged in viruses for gene therapy. You know mRNA can be used as a vaccine, but there are also other genetic treatments on the horizon for that. Antibodies and different proteins are one of the oldest kinds of biopharmaceutical product, and they can be used to replace a bad protein or bind to a bad protein that the body, to protect the body from things like cancer. We also can engineer biochemical pathways or biofuel production in different strains. And one of the fastest growing therapeutic areas is with stem cells in regenerative medicine, ultimately with the goal of growing new tissues for organ replacement or injury repair. But you might ask, how do all these things, what's in common here? And at the end of the day, each of these are produced by growing lots of cells. The cells become the factory for the genetically engineered material to produce or make more of the items in this list. So they all have in common a need for cell production, and that cell production occurs inside of a bioreactor. So one of the main parts of our product line is a laboratory scale bioreactor. You can see two of these bioreactors in the lab setting here. You'll notice there's a small bioreactor vessel connected to a control unit sitting next to it on the bench, and they both tie in to a software system on the adjacent computer. So the controller delivers gases and liquids to the vessel, which help maintain the pH and dissolved oxygen. Heating and cooling is also automated by that controller, and all of the data that the sensors collect, so we're constantly measuring pH, DO, and temperature, that measurement data and the control data is all collected by the computer system, and together these make what we can call a bundle or a complete bioreactor process system. So here first, let's take a closer look at the MyControl. It's a control system that runs small bioreactors, 0.25 liters up to 3 liters. It is our smallest control system, but it's very dynamic, and it has made it well positioned in our market because its small footprint is very easy to fit into almost any lab, but it's still very flexible to run different types of experiments. You can see on the left side what we call our mini bio, a 250 mil glass bioreactor, and on the right side is a 3-liter single-use bioreactor. Besides choosing vessel type, we can also configure parts of the control system as well to meet different customer applications. Remember, we might be serving one customer that's growing yeast and a different customer growing mammalian cell line. So the fact that we can provide equipment that is configurable along with high quality and also easy to use software generates a lot of brand loyalty with our users and is one of our strengths in helping scientists to achieve their goals. But speaking of goals, scientists running experiments in a bioreactor very rarely do just one of those experiments at a time. In fact, we'll commonly see that they'll have a bank of bioreactors that are pictured here, and they use that group of bioreactors to explore more experimental conditions at once. This can often be called screening, where they're testing different new engineered strains. They're screening them against each other, or maybe they're trying to optimize the bioprocess control by changing the pH control point or the temperature. And these preliminary results obtained from the small bioreactor are then collated into some knowledge that contributes to a preclinical molecule or something that's ready to be tested at a larger scale. still at the lab scale. But then we have this controller, which we've just launched, called the Livit Flex. You can see this system is larger and it can operate either 2 to 20 liter glass autoclavable bioreactors or 3 to 15 liter single-use bioreactors. And you can also see that it has two at once. So this system could be a single controller or run as a dual. It's very configurable as well. So it's adapting this controller to the customer application, both in production and in the field. We can also retrofit it easily or use the software to assign the components to a different experiment. So this 2 to 20 liter bioreactor scale at the bench is a very powerful workhorse. We see very commonly that 3 liter is where a lot of process development happens. And so using the gas additions and using the liquid additions to get tight control over the pH and dissolved oxygen inside that bioreactor. We've also implemented smart sensors, so you can connect a huge variety of sensors through a digital integration to this controller, meaning there's data coming with the sensors, and there's essentially a plug and play functionality to make it as easy as possible for the scientists to start up a new experiment without worrying about whether they plugged in the correct sensor. Of course, this comes with modern software, with data logging, and a really dynamic interface so they can see exactly what they want to see to get the data out of that experiment quickly. The other thing I wanted to highlight for you as one of our latest innovations is the single-use vessel that I had shown in a couple of the slides before this. We call that vessel the Apliflex ST. And what's really novel about it is that we 3D print the head plate. So we really can design this single-use vessel for each customer. You can see here we have a 3D printing partner, and we design the most efficient way to print a block of these vessels all at once. And once these come out of that block, they're assembled with the actual plastic vessel itself and the tubings connected, and they get sterilized together. So at the end, we're delivering a ready-to-use, single-use consumable that's in a package that's already sterilized, and people can take these straight to a bench to get their experiments going as quickly as possible, reducing any operator error in preparing the vessel or cleaning, and just making it much more efficient in reducing risk for our end users. I mentioned the fact that it's 3D printed, and what's really cool about that customization is you can try new designs that weren't readily available in bioprocess before. So we have on the left a marine impeller, which is a way of stirring gently. It's just commonly used for stem cells or mammalian cell types where the cells are somewhat fragile. The second one in, called a Russian impeller, is a very vigorous mixing impeller that's used commonly with microbial cell types that are strong and robust and can withstand very high mixing forces. Those are the two standards that the industry relies on, but with 3D printing, we can offer something different, like the helical impeller, which has, these very broad stir arms which sweep through a lot of liquid but still stir very gently. So that enables better mixing without stress on some things like stem cells or other adherent cell types that might be growing on microcarriers. There's also an impeller type called a hydrofoil that might be more suitable for other applications. So that's just one example of something that could be customized on the vessel, but you see the whole white head plate could be 3D printed to add different ports or different fittings that suit the customer application. The other thing we're offering in the Apliflex is 3 different size vessels. So we have a 500 ml, a 3 liter, and a 15 liter now, which we've just introduced this year, which gives our customers the ability to take advantage of everything that single-use offers and also carry out their scale-up studies as they're working through experiments and becoming closer and closer to developing that final process that they'll take into preclinical work, for example. Once you get to that stage, it's common to work in something called a pilot scale bioreactor. And that you can see here from 30 liter to 140 liter, we can provide vessels that could work either on a bench or on their own skid. And probably you notice right away these bioreactors look different. They're stainless steel vessels, so they're sterilized by steam piped directly to this vessel at a bench or on its skid. So nothing goes to a autoclave at a central location in the lab. In fact, the steam is brought to these systems. So they are steamed in place to sterilize them. And although they look quite different on the outside, our engineers have implemented geometric similarity of the interior of the vessel dimensions. So it makes it quite simple from our customers to go from the bench scale, lab scale bioreactor into this scale bioreactor using that geometric similarity. With a bioprocess that has been optimized, let's say perhaps a clinical trial has been approved, the customer might still need more material than they can generate in this pilot scale. And so it is possible to go even to larger vessels from there. You see here some production systems. It might still occur at something like a 20-liter scale for GMP manufacturing, but we can also go up as large as 5,000 liter or put several tanks in series, as you might see here. So these are pieces of equipment being used for GMP manufacturing of vaccines. But we, again, are trying to adapt to the customer. So in our GMP stainless steel systems, the customer specification leads the project. And our engineering expertise allows us to provide different options for the exact production system that the end user wants. So we can change the hardware built into either the vessel or the controller. We can implement a variety of different softwares that are common at industrial scale, such as PLCs like Allen Bradley or Siemens, or a distributed control system like Delta V. And these software systems help automate the processes, such as automation of the steam sterilization cycle on the vessel. And with the Applicon history of making bioreactors at this scale, we're able to bring our expertise, our field service support, application support, and work together to help customers understand what their system can do and ensure that it is producing a high quality product with confidence that they can take to market and put into clinical work. So at the end of the day, the Getinge Life Science Bioprocessing line, we are a full solution provider with the bioreactors starting at research scale and going all the way through process development, pilot, and production. And that set of bioreactors is part of our product line. The bioprocess control is really a key area where we're providing something that works very reliably and automates a lot of the bioreactor process. We are providing analytics, so sensors of a variety of types that integrate directly with the bioprocess control for ease of use, and very powerful software so that our end users can get a better understanding of exactly what occurred during their experiments with historical data, with their current data, and with some tools and templates that make that very easy. And lastly, we also offer something called a perfusion system, which works in very high density cultures that are fully optimized for production of something like an antibody or a protein. Altogether, the Applicon reliable solutions will help our end users in the bioprocess market get to an improved quality of life for their end users. And so we are trying to bring innovation into this space, which provides, the next generation of bioprocess control. We're trying to ensure that that system is configured and optimized exactly to the end user's lab needs so that they're investing in equipment that is specific to what they're trying to accomplish, and they just spend the right amount of money, not more, not less. The systems that we sell are very flexible, and the advanced software functionalities can give them the tools they need to speed up their pharmaceutical or biotech development, bringing that solution faster to market so that we can all benefit. And the intuitive and user-friendly software interface reduces the risk of operator error, it reduces training time, and makes it very easy for everyone to get the work done quickly and save time, so they can do other things as well. And so at the end of the day, modern medicine and technology are producing really fascinating outcomes. There are thousands of targets in research where bioreactors are a key step to producing a biomolecule for treating cancer, a new vaccine, addressing genetic diseases, and providing stem cells that might be able to replace damaged cells, in the human body. And on the right side, you can also see there's industrial biotechnology trying to solve environmental and hunger problems with enzymes, food proteins, nutraceuticals, and bio-based plastics, chemicals, and fuels. So chances are one of these products is already touching your life. And I hope that by showing you how Getinge has supported all of these industries with our bioreactor product line, this is something that you've learned from, and I'm open to any questions if anyone has them. Any microgravity work for space industry? That's a great question. um, Not that I'm aware of directly. I am fortunate to discuss applications with a lot of different customer types, but I don't think anybody that I've worked with recently has asked about microgravity applications. But that's an interesting thought. Great. Well, I really appreciate all your time, and thanks again to the Swedish Chamber of Commerce for hosting this webinar. And I hope that you all have a great afternoon.